Thank you. So already I have already given on this topic. So now I will give some demonstration in this talk. My objective of this talk is to give you some information on whatever the nanomaterials we synthesize and their application for sensing, biosensing, optical sensing, and all drug delivery applications. So before I start, all you know already Dr. Bandana has introduced about me. So I belongs to the CSIR-IMMT as a principal scientist. I'm working there. So this institute is very close to Utkal University, just road across. And this is kind of beautiful institute, a lot of greenery, and we have a lot of facilities. We are working on nanomaterials, different kind of materials, minerals, and a lot of facilities are there. If anybody is interested, they can visit to our institute. I will be happy to host all of you. So this is my group. Some students are working with me in my group. So our group activities, like we are developing some bio and electrochemical active materials, and we conceptualize those materials for development of some bio processes and electrochemical processes for different kind of applications, based mainly on biomedical applications, biosensing applications, sensing, energy conversion, energy storage, all kind of applications used to check with the material we develop. And there are a lot of detailed information. You can find it out from my website here. So this is the outline of my presentation. I will discuss briefly about the introduction of nanoscience and technology. Then I will discuss about this optical sensors, how this nanomaterial can be used for development of optical sensors, then electrochemical biosensors, and their drug delivery is time permits. So let us go to for the introduction of nanoscience and nanotechnology. But this is not a new. A lot of people already, a lot of information are there. So how this, a lot of work has been carried out, how to develop the nanomaterial, and that has been applied in different kind of applications, like you can count from our day-to-day life, from our basic needs also. So that's why, because why this nanoscience and nanotechnology is very powerful, because it has very interesting properties. So that can be gives a sense that we can apply this nanomaterial to solve our problems and solve our purpose. So it is very interesting that nanotechnology is very popular nowadays. You can see just everywhere you turn, or you can go through the advertisement and see this Tata Nano. This is the smallest car in the world. That's why they have coined that word from the nanotechnology, because nano is the smallest material in the world. So that's why it is, and also some of the product like nano complex, Sunsilk also giving hype to the product that they are telling that they have some nano complex. They have synthesized some nano complex because of that nano complex, and that has strong binding to the dandruff, and that removes the dandruff easily and give a robust to the hair. So they are also hyping that product by introducing nano complex. And even Hollywood script also, movie script, there also they are putting some nanotechnology stars, because you can see the Terminator 3 movie, that hero Arnold character, he is telling that he has some arsenal which have some nano trajectors. Because of that presence of that nanostructure, nano trajectors that can control other machines, that can control the bad guy terrorist machine, and they can kill easily. So that's the way what this information is that. So nanotechnology and nanoscience so popular nowadays, and so interesting properties and interesting application. That's why that term has hyped such a label that everywhere now this term has been coined and put to give more hype to the purpose. Even if you can see that nanotechnology language has been introduced, because you can put a nano to any kind of word or any kind of application that will tell us a different language, a different word. You can see the bio applications or biochemistry you are working, you put nano, then nano bio. The nano small particles, dots, you can put nano, then nano dots, nano wires, nano boards, nano devices, like so many terms are putting the nano, and new dictionary words are coming up. So because of such a tremendous application of nanoscience and nanotechnology, you can see this nanotechnology is not only applied in biochemistry, but in different applications, like from energy sectors to get the energy conversion and storage devices, medicines, drug delivery, nanobiotechnology, nano devices, optical engineering, cosmetic industry, nanofabrics. Everywhere there is a lot of application of nanoscience and nanotechnology, because new nano dimension material has been developed for the application of in these directions. If you see the give some example of how this nanomaterial is used for biomedical applications in a drug delivery. That is a tailored morphology, a tailored shape or tailored size of the nanomaterial has been designed so that the targeted place can be hit by taking this drug that can be immobilized on these nanomaterials, and that can be easily delivered to the targeted cell. And also biosensors, like in any kind of defect, any kind of disorder in our body, there is a disorder or a deficiency of some biomolecules in our body that can be easily sensed through development of biosensors that is designed using nanoparticles. Also, nanorobots for brain surgery, cancer therapy using this magnetic nanoparticle Fe2O3 for there is a targeted delivery of this drugs to the cancer affected cells. So that's why a lot of applications there for design of nanomaterial for biomedical applications. And if you can see now a lot of nanomaterial is engineered. You can see major porous silica nanoparticle, those are biocompatible in nature, nanorods, carbon nanotubes, gold nanoparticle which is biocompatible, silver nanoparticle, magnetic nanoparticle, and different quantum dots also that is used for cell line study. And how they have engineered nicely so that the nanoparticle can be properly immobilized on the drugs can be properly immobilized on this nanoparticle, just changing the environment medium, like just changing the pH, that we can release that drug. That can be a lot of work is going on this direction. So we can see this road to nanotechnology. You can see in 1959, this person, the famous physicist like Richard Feynman, who was really dreamed about that. Okay, so time will come, we can develop such a new nanomaterial which dimension will be nanometer scale. So that will give a very interesting subject. It will give a very interesting property, so that all the people will, our society will adapt it, our scientific community will adapt it to change the society, change our applications and systems. So he was dreaming about that, and he has given us such a nice quotation that there is a plenty of room at the bottom. That means you can break down small, bigger particle, you can go down to smaller, to centimeter, to nanometer, micrometer, to nanometer. You will get a lot of interesting property. That means a lot of plenty of room at the bottom. So if you go down to small scales, you will get a lot of different properties. So in 1974, Professor Taniguchi, he has also dreamed about nanotechnology. After the development of these nanomaterials, after a lot of research and getting the knowledge and science of these nanomaterials, we can utilize to develop some engineering. So we can develop some devices that can be done through this nanoscience. And also he has given this term nanotechnology. In 1985, Richard Smell and Robert Call, they have developed and explored that fullerene. This is the smallest nanoparticle, that is C60 bulky ball, carbon. That also got the Nobel Prize. So that is the smallest nanoparticle, carbon nanoparticle. In 1991, also Sumio Iijima, he has developed this carbon nanotube, and 2010 also got the Nobel Prize in development of graphene. Graphene is the very thinnest nanomaterial, so one-dimensional carbon of this graphite-based material. So that's why you can see there is a lot of interesting journey in the nanoscience and nanotechnology. These are the new materials has come, but there is a lot of materials, bulk materials there, that people are trying to develop some new methods how to go down, make it small nanoparticles, and their properties also checking, and they are showing some interesting properties also. So this graphene I have already talked to you. This is a new and interesting nanomaterial so far developed, and this is one atom thick planar sheet of sp2 carbon, that is like benzene. You joined all the benzene molecules that you can get a graphene. It is a more stable 2D nanomaterial. It's a two-dimensional nanomaterial, and you can see this is the mother of all the carbon-based material, because if you stack this graphene, you can get graphite. So if you can take one layer of graphite, that's called graphene. So if you can stack the graphene, then you can generate the graphite, or you can roll it to a cylinder shape so that you can get this one carbon nanotube. Even if you can make a ball, then you can get a fullerene. So that means all the carbon-based material, the mother structure comes from graphene, and the graphene is very interesting property and very interesting application so far has been developed. That's why that got the Nobel Prize. So graphene also has a lot of application in biomedical applications, bio applications in particular, that it is used for tissue engineering, bioimaging also. You can generate some graphene quantum dot, so you can put in the cell line to study the cell structures, and also gene therapy, drug delivery also used, phototherapy, thermal therapy, like a lot of antimaterial applications of graphene oxide has been so far has been developed also. Now a lot of graphene oxide coating and graphene coating has been carried out on the metal substrate to avoid this growth of bacteria for biomedical applications. So just that was just a brief introduction about how we have developed some different nanomaterial, the tremendous applications of her. So now how we synthesize a nanomaterial, I'll just give some brief introduction about that. All you know, still I'm just going to tell about that. That synthesis of nanomaterial is very simple. Either you can regard by two approaches, like top-down approach and bottom-up approach. A bigger particle you can take, you can break it down, you can do the ball milling, or any kind of mechanical force, you can break it down to a small nanomaterial size. Or you can bottom up, you can construct, you can take the molecules, metal ions, then you can do a control reduction. The reduction will generate some metal atoms or any kind of atom. The atom will get nucleated, and the control nucleation, providing some stabilizing agent, structural derating agent, you can able to get the nanomaterials of different shapes and sizes. And this bottom-up approach is quite interesting because you can control the shape and size of the nanomaterial. And also with a narrow size dimension, you can be able to get from that. But in top-down approach, the range of nanoparticle size will be different. And this is all I have already told you. This what about the advantage of top-down approach. Like it's expensive, but some drawbacks at that. Because you can generate a lot of nanoparticles, but it has sometimes drawbacks, like this expensive machine to break down the nanomaterial to nano dimension, and also it's energy consuming process, and a broad range of nanoparticle you can get. Suppose you can break, you can get the microparticle, you can get the nanoparticle. But in case of bottom-up approach, you can do a control assembly, so that it has some advantage, like you can generate synthesized collateral stable nanoparticle. You can do the reproducibility because you can design shape and size, you can able to generate every time, and it's also economically viable because energy is less energy consumption will be there, and you can do the bottom-up assembly. So that is the bottom-up approach, like chemical, photochemical, electrochemical, different approaches are there. Still has already explored that people are working how to synthesize the nanoparticle. So there is three major methods like synthesis of nanomaterial, like this bottom-up approach is like physical method, chemical method, and biological methods. Physical methods like mechanical method, vapor deposition methods. Chemical methods like you can take some salt precursor, you can heat that precursor with some reducing agent or some stabilizing agents. And biological methods, you can do the reduction with some bio plants like plant extracts, enzymes, microorganisms like fungi, yeast, bacteria, any kind of bio-related material you can take it, it can be used to reduce the nanoparticle, you can able to synthesize that material called biological methods, biological process that's called synthesis of nanomaterials. Like you can see this example of bacteria also can reduce this metal precursor to nanomaterials. Fungi, algae, yeast, viruses also. So these are some green way of synthesis because these are not harmful, because the nanomaterial what you will synthesize, these are not toxic in nature. So that's why biological methods has been used, like plant extract, that's good. So you can use that for synthesis of nanomaterial. And this also I have told that plant extract, how that is used, like any kind of plant stem and leaf you can take, you make a powder, you extract some molecules which have the capability to reduce this metal precursor to nanoparticles. So that is again a green synthesis way of producing nanoparticles. So also that's a story about how we can synthesize nanomaterial. It is already well established. But synthesis of spherical shape nanoparticle is well established. People are also established some of the methods, very easy. But the synthesis of different shape of nanoparticle, because already we have understand that you can design a shape, it will be used for like drug delivery applications, and design and tailored size and morphology is very much essential. So that's a different shape of nanoparticles is very much essential because it has some different new properties. And new applications like new optical property, you will see if you can design a different shape, like you can develop a nano rod, it will give a different color and a different like transfers and longitudinal observance. Drug delivery applications, surface enhanced activity, and also high field enhancement effect you can see at the interface between two nanorods. And also you can mimic the nature by engineering some nano hairs, grass kind of structure on a surface, so it will be super hydrophobic in nature. And these are also highly selective for catalytic and electrocatalytic applications. So there's a lot of work is going on. People are trying to synthesize different shape of nanoparticles. So also in our lab, we have also synthesized some of different shape of nanoparticle. We have started with the gold nanoparticle. We could be able to synthesize using the HEPS, like you all know, HEPS is used for a buffer in biological application. We have used that molecule to synthesize some gold flower structures. So that we publish in lung. And also different like nano prisms, pairing tools, we could be able to synthesize using this 5-hydroxytryptamine. That's also very biomolecule that we have used for synthesizing different shape of nanoparticle. And you can see this marigold shape nanoparticle, and you can see marigold shape structures. And these structures is very interesting that we study for surface enhanced Raman spectroscopy, and we could be able to detect some different molecules which has bioimportance molecules. We can do the sensing through surface enhanced by this different structure of this nanoparticle that's called marigold shape nanoparticle that we synthesize using like a 5-hydroxyindoles 3-acetic molecules. And also you have developed synthesized some nanoparticle like using rutin, quercetin, catechol. These are the biomolecules we use to synthesize different shape and the size of nanoparticle. And also you have synthesized silver nanoparticle using rutin as a reducing agent. And this silver nanoparticle has very good, it has shown some interesting antibacterial applications we have started. And also different palladium nanoparticles of different shapes. You can see this dendritic structure and a chain-like structure, porous chain-like structures, we could be able to synthesize. So different metal nanoparticle, even metal oxides we use to synthesize different shape by different things. So that was a story about how we could be able to synthesize some different nanomaterial for different applications. So now I'm going to tell you about how we study the optical applications of nanomaterials, optical sensors. So how this nanomaterial can be used for optical sensing. Like I can show you that this gold nanoparticle, they have interesting optical properties. So how this optical properties comes, you can see this. When this light, you irradiate a light on the gold nanoparticle, but these are metal, but they can show some sort of UV visible spectroscopy study, you can see so they are showing some observance. They observe light at certain UV length, but these are metal. So why they are showing? But usually metal, you understand that metal they don't have any distinct band like homoluma. They don't have, but they are this is continuous. This electron band is continuous. So why they are showing some observance property? The observance property is due to the surface plasma resonance property. How the surface plasma resonance works? When you irradiate a light on the gold nanoparticle, you can see this light when approaches nanoparticle surface, then polarization of the surface charges takes place. You can see this polarization. The surface electrons will be somehow polarized. That will go to one side and to make it positive, another side to make it negative. So that polarization takes place because of this polarization that acts as a kind of molecular behavior, and they are showing some kind of observance that's called surface plasma. So that's why these interesting properties can be tuned by changing the structure. You can synthesize some spherical nanoparticle, you can get only one observance band, but if you can change to different shape, you can get two different bands. Like you can see, this is a nano rod, you can synthesize this light will approach in different direction. It can go through the these directions, it is called transverse, and you can go through the rod, it can call longitudinal. So you can get two observance band, one is transverse band, another is longitudinal observance band. So that also you can see how this light interact with the nanoparticle surface, we can able to get the observance band. So by tuning their size and shape, we could able to monitor, their optical properties, and you can utilize these optical properties for sensing application. That I'm going to give a interesting example, and I can demonstrate here how you utilize this gold nanoparticle for optical sensing application. You can see, you can tell that this gold nanoparticle is a kind of a chameleon behavior, optical properties. So how this gold nanoparticle shows chameleon behavior? You can see here. You see this is a gold. Gold, you can see gold precursor. All you might have see this because of the gold ornament yellow in color, and you can see this gold precursor. We have dissolved gold orochloric acid in water. You can see this yellowish color. Yes or no? But this is gold. Gold, actually, this is the characteristic properties of gold. It will show like yellowish color. Okay. But when you synthesize the gold nanoparticle, you can see, you can able to get this kind of color. This wine red color. Understand? So you can see this property how it changes from yellow to different color. And this observance you can see because of this color, it shows the surface plasma resonance observance that called, and this is showing, you can see. So when how this color depends upon its interparticle distance. You can see this is a different color. If I can synthesize some different way, we can able to get different color. I think if you can take it here, so you can able to understand. One is like deep color, another is fade. Understand? So that means if you monitor the interparticle distance, we could be able to change their observance properties. So their wavelength can be shifted based on the interaction to the light as well as based on the interparticle distance. You can see this gold nanoparticle, it is shows a wine red color. Because of it, the nanoparticle will be dispersed inside the water with some certain distance. But when you can, there are certain distance, you can change their distance. You can change their interparticle distance, that can change to different color. So I have also done some experiment. I will show you how we can do like this. You can see, I have taken this nanoparticle. So now you can see, this is a nanoparticle. This is a pure nanoparticle that I synthesize. So I have added some different molecule that I want to sense. So you can see when I add this molecule, the color has changed. So the interparticle distance between this nanoparticle will be different, will be more. When you add some other molecule, then this interparticle distance is changing, decreasing. So because of this change in the interparticle distance, what is happening? So that changes the color of the nanoparticle, as well as that can be reflected on the change in the observance as well as wavelength. Yeah, so I'm audible to all of you. I just want to show some demonstration because this is some kind of advanced training program. So I'm just going to show some video.
Yes, sir, audible.
Yeah, so just I'm going to demonstrate, to do a demo how you can use for optical sensing. So I will say, I can give you an example like how we use this gold nanoparticle for optical sensing of polyionic drugs, heparin and protamine. So all you know what is the importance of heparin and protamine. All you know, heparin is a anticoagulant and it is used for surgical procedures. And when there is a surgical procedure which takes place, then there should not be any blood clot. So that's why what happened, they used to add some heparin so that can specifically bind to this antithrombin, which is the main responsible for clotting the blood. Yes or no? So when there will be bypass surgery, any kind of surgery, surgical procedures, so that should not be blood clot. Other than what will happen, they will not able to connect these channels, blood channels, the artery, the veins, they cannot able to connect it if there will be a clot. So that's always there should be a blood flow takes place. So for that purpose, what they will do, they will add the heparin so that heparin will bind to the antithrombin. So after this surgery or the surgical procedure will be done, then what they will do, they will test the blood sample, how much heparin is there, then they will add the protamine. So what will happen, the protamine will go and it will specifically and strongly bind the heparin and it will release the antithrombin. Then this antithrombin again will create the blood clot so that the blood flowing will stop and the procedure will be successful. So that's why it has very much importance for heparin and protamine sensing, because it is used for this kind of applications like some surgery applications. So what do we treat? We have tried some lot of different methods are there, but it is hard to, because it's a very big molecules of this heparin and protamine highly positively charged, because it has a lot of amine groups, and heparin is high negative charge. It has a lot of SO3 minus groups are there. So it is very difficult to sense that molecule because a lot of methods, electrochemical method has been tried because this molecule could not able to get oxidized, has some potential. So that's why it is very difficult to sense that molecule. So we have planned to use this gold nanoparticle. How you can use this gold nanoparticle for optical sensing of heparin and protein. So what we did, we have synthesized some gold nanoparticle using sodium citrate as a stabilizing agent. It is very simple. I'm just going to show you how you have synthesized. It's a very few minute experiment. I'm just going to show you, just I'm going to demonstrate it. Very easy. So in front of you, I'm just going to demonstrate. So it is always you have to maintain a safety when you are doing an experiment on nanomaterials, because nanomaterials, all you know, it is some materials are biocompatible in nature, but some materials are highly toxic in nature. So that can, if it go to our body, it will create a lot of diseases and also cancer. So we have to very much careful when you are handling this materials, as well as when you do the experiment in chemistry, we have to be very careful. We have to wear gloves. So what I'm just going to tell you that how we have synthesized the nanoparticle. Just you can see I have taken some water, and I'm just going to add some stabilizing agent. Here this is some water I am taking. So you can see I took some water here. So now I will add some gold precursors. Gold precursors is like this orofluoric acid. That the gold will be in a +3 state here. So I'm just going to reduce that gold +3 to 0. So that gold atoms will generate, and that atom generation can be controlled and can be stabilized by citrate to produce the gold nanoparticle. So here I will use the sodium borohydride as a reducing agent. You can see I'm taking some gold precursors. You can see I'm just adding some gold precursor. You can see here. So I'm adding to the water, and I have some calculated quantity I have to add. You can see this. Okay. So this is some little bit yellowish color. It may not be visible to all of you, but it is. You can see this is looks like transparent, but it's slight yellowish color because I have added some gold precursor to it. So after adding that gold precursor, I will add some sodium citrate, because sodium citrate will help to stabilize nanoparticle, and the sodium citrate will be attached to this gold nanoparticle surface, and this nanoparticle will be negatively charged. So I'm just going to add some sodium citrate. I prepare some concentration of required concentration of sodium citrate. I'm just going to add. You can see I just added here. Yes. Yeah. So you can see I just added here. It's an added. Then now I'm going to add some reducing agent to produce the nanoparticle. You see this is a slight yellowish in color. I just been checking it, but it should be proper styling. But this facility is not there in my office, but it is in my lab. So I'm just going to follow some method. But when you do the synthesis, you have to follow proper way, and you have to do in your fume hood and a proper safety and the precautions you have to take. So now what I'm going to do, I'm just going to synthesize it by adding some reducing agent. So here you can see, Bandhan, I'm audible to all of you? It is visible to all of you?
Yes, sir. It is visible, sir. Visible and audible also.
Okay, thank you, thank you. So here you can see here what I did. I'm just adding some reducing agent. I'm just checking it. So you can see I just checking it, and here I'm adding. You can see then I have to add a little more quantity. You can see there is a change in the color. Then I'm adding to little more, so that I'm just going to set it. Then I have to add a little more. So you can see now, you can see this color is changing to some reddish color, like one red color. I have to add a little more. It should be proper styling because I'm checking that, so it is taking time. So is it visible to all of you? Vandana, it is visible? Hello? Hello. So now you can see this is the color of your gold precursor. So when I have synthesized the gold nanoparticle, you can see this wine red color. So is it visible to all of you? So Vandana, is it visible?
Yes, sir, it is visible. Sir, it is slightly right, sir. Please make it slightly right. Yeah. Now it is visible, sir.
Yes. You can see I'm making it very close. So
Yes,
that
sir.
you can see
Yes, sir. Now it is visible.
this is gold precursor, and when I have synthesized the gold nanoparticle, this is the reddish color. So now you can see, so how you can use this for optical sensing applications. So now I have transferred to some gold nanoparticle to one pen drop. So you can see this is one pen drop, another pen drop. I'm just putting to make it compare. Just a minute. Yeah. So you can see this I have taken with 2 pen drop. So you can see because this pen drop is not transparent that much, so that's why it is looking like this. But in glass, it is really looking good. So you can see this color is like this. So this will give a different observance and the wavelength. But if you add some molecule to it, suppose you want to sense that molecule, how the color is changing. Yes, I'm just going to do one experiment. I'm just going to add some potassium. But already we have done some experiment with potassium. Just I'm going to add some potassium to it. So what will happen? So if we'll add some potassium, so in front of you, I'm going to add just so now you see the difference. So after adding, you see the color is changing. So different color. So that means you see. So it is also very. This is the difference between these two. So you can see it was like one red color. Now it is going to like pink. Some different color it is coming. Yes or no? Some bluish color kind of. So that means you can see I have added the protamine. What the protamine did? It has aggregated it. Aggregated means it has attracted this gold nanoparticle so that this gold interparticle distance has decreased. So that's why the color has changed, and that will can be reflected as a change in the wavelength and as well as observance. Yeah, so that's I'm just going to tell you details on this. So what we did, you can see this is the gold nanoparticle. When we have synthesized the site test and the nanoparticle, when you add protamine, what happens? The protamines are highly positively charged. They attract this gold nanoparticle and they close this gold nanoparticle closer. They make it closer so that what happens, the interaction of light with this nanoparticle environment will be different than the previous. So that's what happened. That has been reflected. You can see this was the color. When we add protamine, the color is changed. But next, what we did the heparin. What happened? The heparin is interacted to the protamine, then it is releases the nanoparticle. Then this color is coming to this direction. But it takes time, usually 2 to 3 hours or 4 hours it will take to again come the original color of gold nanoparticle. So that you can see here when you add the protamine, the observance is changing and the wavelength is also shifting. So that change in the observance as well as the shifting in the wavelengths, that will tell you how much concentration of protamine and heparin we have added. And this is the TM, transmission electron microscope, showing how this nanoparticle was there in the water. Just we put a drop and you take it out from the grid, you can see this nanoparticle are just like this. There are some distance between the nanoparticles. So if you add protamine, you can see that make it closer. Here you can see the image I have shown. So that means they are aggregating this nanoparticle, they are making it close. If you add more quantity of more concentration, then what the aggregation rate is increasing. But when you add heparin, then it is releasing this protamine and it is binding to the protamine and releases the nanoparticle. You can see this interparticle distance is again further increases. But this is not that 100% reversible, because some aggregations are there, but the wavelength somehow it will go to the original position, not exactly the original position, but it can shift to that direction. That means again further intraparticle distance will again increase. So using this interparticle distance monitoring, we could be able to change the color of the gold nanoparticles from wine red to blue, even the different colors based on the concentration. So that's why that we used to tell this gold nanoparticle has a chameleon properties, that you can be able to sense any kind of biomolecules of our interest by using this optical behavior of the gold nanoparticle. So that was a story about how you can use this nanoparticle for optical sensing application. So now I'm going to discuss about the electrochemical application, development of some electrochemical biosensor using these nanoparticles. So before that, let me give some introduction about that, why we need biosensor, because it's very much essential for our health. All you know what the importance of health for us. So how you always don't care about your health by using some different kind of bad food habit and a lot of other kind of pressures in job, stress, a lot of things are there that can be reflected as a disease or sickness in our body, and that's why we have to go to the doctor to understand what happened to us. So when we go to the clinic, then we have to do some sensing. They will prescribe that you go for test of this blood, this urea, this, that. So then detection of clinically relevant molecule will be essential when you feel sickness or disease, to understand what happened to our body. There is a fundamental understanding of their biological and physical function of this clinically relevant molecule. Suppose if there is a change in the glucose concentration in our body, that is called diabetes. So that we should understand how much glucose is there in blood sample, how much urea is there, how much ascorbic acid is there, how much like any neurotransmitters when there is a disorder in mental disorders, they can sense how much neurotransmitters are released. So that kind of that means there is some clinically relevant molecules are there in our body. We have to sense that how much concentration is there. So that's why you have to find a device. You have to find a sensing system to understand how much exact concentration of this clinically relevant molecules are there in our body. So you can see this is a chemical input paper. It's showing there's plenty of molecules, like their concentration in our body varies. Some molecules are having very low concentrations, like femtogram per liter to picogram per liter, nanogram per liter. Also, some molecules are micrograms, milligrams, grams, like they are in our body. So they have a fixed concentration. As a healthy person, they have some fixed concentration, a range of concentration there. So if there is a change in the concentration, that will reflect any kind of disease or sickness in our body. So these are some kind of molecules like neurotransmitters, metabolites, vitamins, amino acids, and like that. So you have to develop some kind of biosensing system to understand those clinically relevant molecules. So how we can be able to sense that molecules? So you can develop a biosensor. So how a biosensor works, you can see you want to do the detection, or you want to monitor, you want to sense these analytes of our interest. For that purpose, what we have to do, we have to design a bioreceptor. Bioreceptor that have the property that can specifically interact these analytes. Like enzyme, that is a specific interaction of some of the analytes. Suppose you want to monitor some like lactate. So lactate dehydrogenase is some enzyme that will only interact with the lactate. Similar, like alcohol, you want to do sense. So alcohol dehydrogenase you have to take. Those glucose you want to monitor. Glucose oxidized enzyme is there that have some specific interest in glucose. So like similar, you can take enzymes, or some antibodies are there, microorganisms are there, and even some materials you can design that when the analyte will interact, that will give a change in the current signals and resistance signals. So that through that change, we could be able to find some kind of signals. So that interaction will give some kind of changes in the kind of pH change, heat change, light, mass changes, some electroactive substance change. So that can be sensed with a transducer. So a transducer means one electrode that will be that can accommodate this bioreceptor on the surface. When an analyte interacts with this bioreceptor, that will give some kind of changes on the transducer. So that changes, that signal, change in the signal can be measured using some kind of devices. So that is a kind of, these are the general component of a biosensor. So you can see, put analyte, put a bioreceptor, then that will be accommodated on transducer. When the analyte will interact with the bioreceptor, this transducer will give some change in the signal. That signal can be converted to electrical signal, so that can be monitored through ear devices or display devices and detectors. So that's different kind of biosensor based on the property of materials, based on the property of a bioreceptor, based on the property of analytes. So far has been designed. There's a bioluminescent biosensor, electrochemical biosensor, optoelectric devices, piezoelectric, thermistors, like that. So out of that, some electrochemical devices has also a lot of applications, and so many electrochemical biosensors so far developed. Like that also different varieties like amperometric, change in the current, conductometry, change in the potential, potentiometry, also there is a change in the potential, measuring the superficial charge like a quartz crystal microbalance is there. So suppose you put a molecule on this surface, that will give you some kind of change in the resistance, change in the impedance. So that we can monitor through electrochemical. That's called electrochemical biosensors. So there is a lot of applications of biosensor, not only clinical point of view, like some molecules there in our body that we could be able to sense through a biosensor. From clinical point of view, like in vivo or in vitro, we can directly monitor some of the molecules by inserting a biosensor to a living organism, or in vitro you can take out these concentrations and do in your clinic. And non-clinicals like any kind of environmental, food ripening, and pollution monitoring also can be done through some biosensors. So that's all you know. There's a lot of history so far. So many different way of biosensor has been developed. There is a lot of modifications, improvement as so far has been carried out. And I'm just going to give you an example of glucose biosensor. So 1962, Professor Clark, he has designed a glucose biosensor in 1962. But so far there's a lot of development to measure a glucose in our body. So because that measurement, because that time what he has developed, that could be able to sense, but that improvement was not there. That electrode was not stable. That sometimes the electrode get foul, and not recycle cannot be done. That's why there's a lot of improvement has been carried out. That's why they have given like first generation biosensor, second generation biosensor, third generation biosensor. Just like our automobile vehicles, you can see different kind of vehicles based on the requirement and based on the environment we are developing. Similarly, there is a lot of steps of improvement of the biosensor has been developed. Actually, what type of ideal biosensor? Why there is an improvement? Because so far no ideal biosensor exist. Because to improve, to meet the criteria of ideal biosensor, a lot of improvement is going on. So like sufficient accuracy and repeatability. So you have to accurately that okay, I have to detect like 1 nanomole concentration. That should be sufficiently accurate. And also repeatability. Every time you can sense that 1 nanomole. Sufficient speed of response. You dip the biosensor, quickly get the result. So if you wait for a few hours, that's not good. And sufficient dynamic change, that means if other molecules are there, it changes that can be reflected through this only. And also insensitive to the environmental interface and some other, like there's a lot of criteria properties, not this only this much. A lot of criteria is there for to develop an ideal biosensor. To meet that properties, a lot of improvement has been carried out. Like in electrochemical biosensor, a lot of biosensors have been developed based on that high sensitivity. Electrochemical biosensor has high sensitivity because this is related to the current signal. And the current, if you can get nanomolar, you can able to measure picomolar current. We could able, nowadays we have a lot of different devices. We could able to sense like nanoamperes, picoamperes currents. So that is related to very low concentrations of some different molecules, and also highly selective. Some molecules have, they can oxidize some particular oxidation potential, so that interference will not take place. So it will be highly selective, very low detection. As I have already told that very low concentration we could be able to detect, and also rapidly we can monitor. And through channel of electrode, like 20 channels of electrode, 40 channels of electrode, by simultaneously you could be able to monitor some different like 20 or 10 different clinically relevant molecules. So I'm just going to tell about this electrochemical biosensor. So now how this electrochemical biosensor has been designed. So this is very simple. This is a device contains a biological recognition element like enzymes or any kind of bioreceptor like proteins, antibodies, nucleic acid that we put on the electrode surface. And when an analyte is approached to this bioreceptor, that gives some electrical signal to the transducer, and that transducer provides the change in the concentration of the analyte can be reflected in the change in the current. You can see this current is directly proportional to the concentration of analyte that interacted to this bioreceptor. So from this current, how much current you are getting from their interaction, that will tell you how much analyte it is interacting. So it's very simple. So already I told you, so that electrochemical biosensor are different types like biocatalytic sensors. When you use the enzymes, when some molecule you add, that get oxidized, that get reduced, and give some oxidation current and reduction current, that's called biocatalytic. And it catalyzes the process. And affinity, just like interaction, just a molecule go and interact, also adsorb or bind to the another molecule. That binding will give some kind of changes on the electrode surface. That is called affinity-based biosensor. Another is biocatalytic-based biosensor, where enzyme is used. Already I told you that different biosensor has been developed, electrochemical biosensor. That's called first generation biosensor. When Professor Clark has developed, what he did, he have taken some glucose oxidase inside the solution. So inside, and they put a conducting electrode inside the solution. What happens? Any analyte suppose interact to the enzyme, and it is getting enzymatically converted to product, and in the presence of oxygen, hydrogen peroxide is generated. Then the hydrogen peroxide comes to the electrode surface, get oxidized and give electron. But that process was very slow, because in the solution, enzyme will convert to the analyte to product, and hydrogen peroxide will generate. Then that hydrogen peroxide will go to the electrode surface. But how much hydrogen peroxide generated, it is very difficult to monitor that time. So this reproducibility and the sensitivity was not there. So that was called first generation biosensor. Further, some second generation developed where they have used the redox mediator to catalyze this process very fast, like hydrogen peroxide is generated. So quickly hydrogen peroxide is not coming to the electrode surface. That what they will do, they have used some redox mediator. So when enzyme is oxidize some molecule, and the presence, it gives the electron to some redox mediator, and redox mediator will get quickly reduced. So that cycle will take place very quickly if you use redox mediator. So that time what happened? So somehow the sensitivity increases and the speed of monitoring is increases. They give you a second generation. Further, there is a loss of concentration, loss of signal. So that's why further what they did, they just accommodate this enzyme on the electrode surface. They don't put the electrode inside the solution. And this redox mediator, they are very nice sensitivity, and after a few hours, they also get decomposed. So to avoid that, they put direct enzyme on the electrode surface by putting some kind of mechanism so that this enzyme activity remains, because enzymes are very sensitive, they can decompose, denaturize. So that's why what they put direct enzyme on the electrode surface. They call it a third generation biosensor. So now they are accommodating nanoparticles, enzymes on the electrode line, and fourth generation biosensor that I'm going to talk about that. So when this electrochemical biosensor is designed, so based on the biocatalytic biosensor, one is like enzymes is used. So that's a different kind of enzymes are. One is oxidase-based biosensor, where you can see this is oxidase-based enzyme is used, where the substrate is your oxygen. So in the presence of oxygen, this enzyme will work. If you put an analyte, this enzyme will be converted to its product in the presence of oxygen, where hydrogen peroxide is generated. That's called oxidase-based biosensor. Suppose you want to monitor, you want to understand the concentration of this analyte or substrate, you have to monitor how much hydrogen peroxide is generated. So this generated hydrogen peroxide concentration is directly proportional to the how much the analyte is interacted to the enzyme. So that's why you have to develop some system which can effectively monitor the enzymatically generated hydrogen peroxide. That is based on oxidase based biosensor. So another is dehydrogenase based biosensor. The mechanism is very simple and similar, but here the cofactor is your NAD plus. Here it was oxygen, it is NAD plus. NAD plus will be enzymatically converted to NADH when an analyte is interacted to NAD. It is a dehydrogenase enzyme and that generated NADH. If you can understand how much NADH is generated, that you can tell you how much analyte is interacted to the enzyme. So these two enzymes, this biosensor is developed based on its efficiency towards hydrogen peroxide when you use the oxidase-based enzymes. When it is effective to NADH, then that time you have to do some dehydrogen-based enzymes. So that's why the mechanism is different. So that's why the electron which has been designed, that response to hydrogen peroxide will be different than the NADH. So that is separate electrode, separate material, separate property of material is designed so that it can be effectively monitor hydrogen peroxide as well as NADH. So when we started our work, you know this is a structure of NADH, that is a electrochemically 2 electron 1 proton oxidation reduction takes place. Then we try to develop some nanoparticle modified electrode for first to understand how it is in response to NADH. If we showing NADH response, then we can use to develop a dehydrogenase based biosensor. So then after modifying this gold nanoparticle on the electrode surface, and it is very simple and a very easy way, we have used some Mercator profile trimethoxysilane. That is some kind of silicate network having thiol groups, 3-dimensional network we put on the gold electrode, and because of these thiol groups, we just self-assembly. We dip this electrode into the gold nanoparticle. This nanoparticle is self-assembled to the thiol groups, because gold thiol has a specific interaction. That's why it is properly self-assembled on the electrode surface. So after modifying the gold nanoparticle, what we observe that is you can see this is the bulk electrode. You can see this in response towards the NADH is using, this is not showing some prominent peak, and it is oxidized very higher over potential, like 0.9 volt. That means you have to give more potential, more enzyme energy you have to consume. But when you modify to gold nanoparticle, it is oxidized at lower over potentials. So that's why this gold nanoparticle modified electrode shows good response towards NADH. Further, what we did, we have developed some amperometric method, because this is a very selective and electrochemical technique where its technique is very highly sensitive. Sensitive in the sense very low current you could able to measure, like nanoampere current, picoampere current, so that you could be able to detect very low concentration of NADH. So that if enzymatically any analyte interact with the dehydrogenase enzyme, very low concentration, then very low concentration NADH will generate. If you can monitor that concentration, then it is good for. So that's why this technique, like amperometric technique, is used. We could be able to monitor 5 nanomole concentration of NADH. So that is a very low concentration. And also, we study a range up to what concentration of NADH could be generated. Then we try to fabricate a biosensor by integrating these enzymes on the electrode surface as well as nanomaterial on the electrode. Both are there. So both role is there. So this both are where on the electrode surface. This is a 4th generation biosensor where we suppose this is your analyte. You can say it's a substrate. When you add to this solution, what happens? It is enzymatically converted to product, and it will generate in the presence of NAD plus NADH. And this NADH, we have observed that gold nanoparticle is good. It can easily oxidize this NADH and provide the electron to the electrode surface that can be reflected, increase in the current. So when you add this analyte, and we have observed that there is an increase in the current was observed, that we use for sensing of ethanol, sensing of lactate, also using lactate dehydrogenase, and ethanol using alcohol dehydrogenase. Okay, so by using this effective electrochemical biosensor, you could be able to monitor those concentrations. So that was a story about how you can be able to design a dehydrogenase based biosensor. But sometimes some material they don't have response to us NADH. That time what you have to do, you have to take some oxidase based enzymes. So that's why you have to develop some different other material which you have shows some good response to hydrogen peroxide. So here I'm just going to show some example of how you design some oxidase based enzyme, biosensor. This is oxidase enzymes. And when you add this analyte, what happens in the presence of oxygen? It generates hydrogen peroxide. That hydrogen peroxide is getting oxidized on the electrode surface and provide the electron. So for that purpose, what we checked that platinum nanoparticle shows better behavior towards hydrogen peroxide. We also immobilize this platinum nanoparticle on the electrode surface. After immobilization, we have observed that nanoparticles is successfully immobilized on the electrode surface. Then what we did? We just checked how its response toward hydrogen peroxide, and we have checked that it oxidized hydrogen peroxide at some this potential like 0.4 volt, 0.45 volt, and then further I have already told you that we have used amperometric technique because this is very sensitive technique. So with this technique, we could able to monitor the concentration of hydrogen peroxide up to sub-nanomolar, like 0.1 nanomolar, and highly sensitive also. So as this nanoparticle particularly modified electrode shows high sensitivity towards hydrogen peroxide, then we try to decorate. We try to design a biosensor by putting this oxidase enzymes and this platinum nanoparticle on the electrode surface. And you use this nanoparticle for the sensing of uric acids. Uric acid. We have taken the uric acid, and you can be able to see. And we also developed a patent on this work. And this biosensor was really. So with this, we could be able to develop some kind of biosensors. So another biosensor we developed, that is I am going to show you how we have used the graphene. So Dr. Pradhan, are you there? So let me take some 1 minute break. I will take some water.
Yes, it is.
Yes. Okay. So next, further what we did, we have used of graphene. As I have already told you that graphene has some lot of biomedical applications, biosensing applications. So why there is a use of graphene? So we have seen that there is a lot of advantage when you use the graphene. So we have developed some electrochemical biosensor. Here also we have developed some electrochemical biosensor using the graphene as a conducting substrate where we accommodate them some oxidase enzymes and some platinum nanoparticle on its surface. And it shows better property that I'm going to discuss. Here we try to design some electrochemical biosensor by integrating this oxidase enzymes and platinum branch structure, some different structure on the graphene surface. And we use for biosensing of xanthine. So first what we did, we try to understand how its response, its platinum nanoparticle on the graphene is response to hydrogen peroxide. What we did, we have developed a method to synthesize some blanched nanostructure. You know, I have already given an introduction that when you change the shape of nanoparticle, their property and their interaction to the molecule is different. Their property is enhanced. So when you use some branch of tiny nanoparticles of branch structure rather than spherical, its interaction to the hydrogen peroxide is really good. So when we decorated this platinum nano branch structure on graphene, we have observed that it could able to oxidize this hydrogen peroxide at 0.37 volt. But previous, when you use the spherical platinum nanoparticle, it oxidizes at 0.45 volt. That means it is reducing at around 1.1 volt. So that means it is also less consumption of energy. So also we observe that when this peak position, peak current is good and current density what we got is better than that previous work. So then what we did, we checked how it's a response to different scan rate, whether the scan rate has some influence and also it is whether it is a diffusion control process, because we need diffusion is essential because the hydrogen peroxide generated it will diffuse to the electrode surface. And further we checked with the cyclic voltammetry to understand the change in the concentration. When you increase the concentration of hydrogen peroxide, you see the peak current is increasing. So that is directly proportional to the current we got from the oxidation of hydrogen peroxide. Then what we further we have done the amperometric sensing, And through the amperometric sensing, we could be able to monitor the concentration of hydrogen peroxide. Here also, it is showing a amperometric sensing to 5 nanomole, but we could be able to detect sub-nanomolar concentration of hydrogen peroxide by this graphene and platinum nanostructures. And also we studied the stability. Suppose you try to develop some electrode, whether that is stable for a longer time or not, and also that's operational and storage stability. Some technical system is used to evaluate the stability. Like one is operational, means you do like continuous measurement. So every time you do, whether that current we are getting similar or not. And storage stability, today you have done use the SPI sensor, then store in a like any phosphate buffer or any kind of ambient atmosphere you store. Then tomorrow also you check after 24 hours whether that also showing the reproduction or not. That we have shown some storage stability and operational stability. We have observed that this is up to 14 days it was stable. Storage stability and also operational stability, you can go for like 1 hour. The current remains steady and maybe around 20 minutes or maybe around 40 minutes, this is quite stable. After 1 hour, some decrease is there. That means up to continuous 1 hour we can use. Means every time, like this kind of experiment, we need maybe for 5 minutes to do an experiment. But up to 1 hour we can continuously measure. So that's its storage and operational stability is quite good. And also we have studied the interference. Interference means whether other molecules they are in the biological environment, like in a body sample, ascorbic acid, uric acid, dopamine, glucose, all are will be there. Whether they can come and interact to the hydrogen peroxide, the potential where it is getting oxidized. If they will come and they will oxidize at same potential, then they can give a current because of them. So that will give an interference. That means you will not able to understand whether that current is due to hydrogen peroxide or due to the uric acid. So also we tried to do some experiment that's called interference test. We have checked the amperometric test by adding the hydrogen peroxide, and we observed that there is an increase in the current. But that same experiment, we have added ascorbic acid, uric acid, glucose, dopamine. We have observed that current increase in the current was not there. That means they are not interfering at that potential. So then what we did, we have let us try to fabricated a biosensor we have used for sensing of xanthine. Because xanthine, all you know what is the importance of xanthine. I'm not going to tell all your biomedical or biological applications are there. So xanthine is also used to detect the freshness in fish or meat, because if a meat and will decompose or fish will decompose, then xanthine will generate. And that's why the storage purpose of this fish and meat, we have to develop sensor, we have to insert the sensor into the package without damaging the package. If a lot of xanthine is generated, that means it is decomposed. We have to throw it down. So that storage of this fish and meat, it is very much essential. Another also that xanthine has some kind of role. There is a very clinically significant diseases is caused because of the lack of xanthine, that is called xanthinuria or Lesnian syndrome kind of diseases. So that means what I'm going to tell you, that we collected from literature, that xanthine sensing or xanthine detection is very much important. So what we did, we have used some xanthine oxidase using this graphene and platinum nanoparticle modified electrode. We have decorated on its surface both xanthine and platinum brass nanoparticle. And when you introduce the xanthine, then what happens? The xanthine interact with the xanthine oxidase, then convert to a product and generated hydrogen peroxide. That generated hydrogen peroxide get oxidized by this brass platinum nanoparticle on the graphene, and it gives the current signal. And that signal is directly proportional to the concentration of xanthine used. So with that, we could be able to sense the concentration of xanthine. So that was the story how we have developed some electrochemical biosensors by using some enzymes and oxidase-based enzyme, oxidase dehydrogenase-based enzyme, and that how the nanoparticle helped for the sensing of this clinical relevant molecules. And some other molecules like non-clinical relevant molecules, but they have importance. that also yeah, that molecules are highly contaminated. It's like toxic in nature. So that will create a lot of diseases in our body. So we have to sense that molecules. Also for that purpose, we have designed some this nanoparticle modified electrodes. First, we are going to discuss about how you use this nanoparticle modified electrode for sensing of arsenic. This arsenic, mercury, lead, cadmium, these are chromium, these are highly toxic elements, and that is used, and that is some of the molecules are there. Sorry, some of the elements are there in our air crust. That is some ores are there, so that get contaminates our drinking water. But those molecules, those elements are highly toxic in nature, so that creates a lot of diseases. One example is there like arsenic. Another example is mercury. These are highly toxic and they are associated with many health problems. One is you can see is arsenic. When it will be associated to, you can take the arsenic contaminated water every day. So then what will it will going to create a kind of different diseases like you can see this lung cancer, keratosis, skin lesions, like you can see some of the people in this arsenic contaminated area. Those are living there and taking this arsenic contaminated water. You can see there is a lot of roots in their palm and their foot. You can see. So that's called skin lesions. So that's why this arsenic is highly deadly in nature. Because they're highly toxic in nature. But this is a slow poisoning. Not all of a sudden it will kill. But after 10 years, that will cause cancer and skin lesions, and then the person will die. So that's why the World Health Organization has given provided a guideline value that if you less than 10 ppb is there in drinking water, then it is safe to drink. Other than it will create this kind of serious diseases. And similar US Environmental Protection Agency, EPA also further review that case, and they have observed that okay, 220 ppb means even if more than that also create a danger. So like it is very safe if you can detect arsenic, you can arsenic concentration lower than 2, then that is really safe. That will not cause any disease. So that's why what I'm going to tell you that because of this toxic element, if it's present in our drinking water, that is going to create a diseases. So that's why we have to develop some kind of sensor which can monitor the arsenic, how much concentration there in our drinking water. For that purpose, what we did, we tried to modify with gold nanoparticle on this electrode, and directly we have taken arsenic, and we have observed that arsenic is, we have used some square voltammetry technique. We hold some potential so that arsenic is getting deposited on the electrode. Then we have done the stripping mechanism. That's called stripping. That means again further oxidized, so that the arsenic what is present on the electrode surface it get oxidized. From there stripping mechanism, you could be able to understand how much arsenic is interacted to the electrode surface, and that will give you some current signal. So for this method, we could be able to solve ppb. You can see 0.02 ppb. You can see how sensitive this nanoparticle modified electrode. Even if you can, you see 2 ppb, 20 ppb is safe, but before that also it could be able to monitor. So that means it's highly sensitive, and that can be used for monitoring arsenic. So and also you checked also mercury, it is also oxidizing mercury and different potential, and so sense the mercury, and some copper also doesn't interfere at this potential. Then after successful doing the sensing of this arsenic in the lab, we try to collect some real sample. So if some area that is affected with arsenic, how much concentration there, because real environment will be different what we are working in the lab. So that's why you find some literature. We have observed that some parts of Bangladesh, India, and China, this Asia region I'm talking about, that's also affected with arsenic. And our India, like in West Bengal of India, that's like 3,417 villages, somehow some percentage of arsenic contamination are there, because that geographical region is like that. There is a lot of arsenic ores is there inside the earth. So when they this we can dig a well or tube well, then what will happen? The water what is present in the groundwater also contaminated because of the presence of ore. So that's why the groundwater is already contaminated, and when you take this water, it is also toxic because arsenic will be there. So we have collected some water from this Barakpur district, I think block 24th Parganas district. some villages like Nadia district, and there I collected some Bharat block, some samples, and you can see it's the map showing the red regions where the arsenic concentration is more than 50 ppb. So we have collected some of these samples from these red affected regions. So you can see, so we have collected real sample and we have checked and we have monitored that it has around 51.4 ppb. So we, according to the literature, and it was like 50, but we could able to like 51. This pair is like somewhere must be there. So that means our sensor shows very good and it's effective for monitoring real sample analysis, and we have done a patent on this. So that was a story how we utilize this nanoparticle modified electrode for sensing of this some environmental contaminants. Those have some kind of impact to our health and they can cause a disease. So we need a system to monitor their concentration before we can utilize them. So another thing I'm going to tell you how we develop some electrochemical biosensor for neurotransmitters. So all you know now, neurotransmitter, how important it is for us, how they control that, our neurotransmitter controls our activity, our emotions, our thinking, yes or no? So dopamine is one of the neurotransmitter, one molecule is there. So this has some clinical element like our feelings is controlled by dopamine functions, like feeling of pleasure, bliss, or you are feeling some sad. And if there is a dopamine deficiency, you feel sad, and you will feel bliss if there is a actual concentration of dopamine there. So that's why there's some psychiatric patients, those are feeling depressions, that are very sad. Then there is, that means that there is no proper release of dopamine takes place. So we have to understand how much concentrated dopamine is released from the brain cells. We'll monitor, and accordingly you have to give some drug to enhance this release and to increase the concentration of dopamine. So for that purpose, we try to develop some micro electrode. Because why we develop micro electrode? Because bigger electrode we can insert to our brain cells so that you can in vivo you can be able to monitor. And also in vitro, one cell is like that PC12 cell. Those are cells which are responsible to release the dopamine. So the cell structures like microdimension. If you put an electrode and cover the cell, then how much the cell releases through exocytosis of dopamine, then that we can able to monitor. That's why we have fabricated some micro electrodes, like say for example 5 to 10 micrometer dimension carbon fiber micro electrodes, and then we have etched out some carbons and we have deposited gold, then we polished it and we have modified this gold nanoparticle on these fibers, so that this gold nanoparticle modified electrode shows very good response to the dopamine. And then we have sensed the dopamine, and you can see without this gold nanoparticle, here the picture shows how sensitive when you add the gold nanoparticle. This gold nanoparticle is highly sensitive to dopamine oxidations, so we can get some high current signals. So from this current signal, we could be able to understand how much concentration of dopamine is released by these cells. So based on that, you can prescribe the drug to increase the concentration of dopamine or decrease. So that is the importance of how you use the gold nanoparticle modified electrode for the electrochemical sensing of neurotransmitters. So that is also we have developed some different kind of electrochemical sensors like now enzyme free. Sometimes enzyme, you know how delicate it is used. You can develop a sensor without using enzyme and it can effectively monitor the concentration that will go. We can be able to monitor glucose, chromium 6 also. Chromium 6 is highly toxic in nature. That will be developed, and ascorbic acid, isoniazide, that's also another drug used. So that we could able to develop some electrodes for the sensing of these molecules of applications. So these are a lot of work is going on. Whatever the biosensor you have to develop, you have to develop. This is a need of that means we have to develop some kind of biosensor which have a lot of application like food analysis, drug development, crime detection. So some of the molecules are there that is used to do the crime, and some forensic science sensing also. We have to use some electrochemical biosensor, environmental monitoring, medical diagnosis. A lot of applications of electrochemical biosensors there. So you can generate some good nanoparticles whether you can increase the sensitivity, increase the selectivity, so that will be good. So that's a lot of work is going on in this direction, and nanoparticle has a lot of application. Another, I am just going to tell you how we have developed some different material for bioimaging and cell viability study applications, and also anti-cancer drug delivery. Some of the nanomaterials are very good that is used for bioimaging application. Suppose you have given some drug to the cell and that cell get collapse, like suppose cancer cells get die. So how you will understand? You have to do some cell imaging study to understand whether that cell is getting killed or with that drug or not. So that for that purpose, so some bioimaging purpose, we have to develop some nanomaterial, so some optical property must be there. So when you put the light, you can see there is a change in the color. And also that nanomaterial should have cell viability assay study, because that means that should not damage to the normal health cells. So you have to study whether they are biocompatible in nature, they should not damage to the healthy cells, and they should not and they should kill to the other affected like cancerous cells. So in that purpose, we have contributed by developing some kind of carbon nanodots. In our lab, we have synthesized the carbon quantum dot by using very small dimension carbon molecules. So what we did, we have taken some carbon precursors like HEPS. That's a very good biomolecule. And what we did, we have synthesized some hydrothermal method. What we did, we have taken the HEPS and put in the hydrothermal cell. Then we have increased elevated the temperature so that this hydrolysis and condensation of this molecule takes place, so that will form a nanoparticles of carbons. When this nanoparticle of carbon form, that is below 10 nanometer or below 5 nanometer, that shows some quantum behavior. When this light is interacting, there is some kind of band changes takes place, and that will give some optical behavior. Photoluminescence property you can see, and when you irradiate it with UV light, you can see some greenish color or bluish color that develops. And we have done a lot of characterization how to the structure of this carbon nanodots. It is very small nanostructures, less than 10 nanometer. And after successful synthesizing these carbon nanodots, what we did, we have used this carbon nanodot for anti-cancer drug delivery applications. So we have used some kind of drug, anti-cancer drug, you immobilize on the this carbon surface, and we have delivered this carbon surface. This drug immobilize the nanoparticle to the cancer cells that I'm going to tell. So before that, before we study the drug delivery test, so this is a work we did in ILS with my collaborator, Dr.Debasish, and we have synthesized this carbon dots using HEPS buffer, and he has studied this cell viability assay, and it has observed that he has taken some HEC-293H65 cells and treated with this 24-48 hours. And it was observed that it has successfully tested the cell viability assay, but it is not killing these health cells. And also he has also seen the bioimaging. When you put the nanoparticle into the cell, you can see that cell can be able to detect through this confocal microscope where this nano rods has accumulated into the cell, and there when they put the confocal microscope, and they could able to see the color. Then after that, what we did, we have immobilized one drug like anti-cancer drug, doxorubicin. So doxorubicin, all I know that is anti-cancer drug. So that we immobilize, which is some kind of chemistry we did in our lab, and we have studied that pH change release. If you can change the pH, then drug will release, and if you can increase the pH, the drug will release. So based on the environment of cell, inside the cell and outside the cell, when a drug will interact, this nanoparticle molecular drug will interact with the cell surface, so that the cell to release this drug to the cell. So what we did, you can see this HEPS method used for synthesis of this carbon nanodots. And after synthesis of this one, we have immobilized doxorubicins and with some kind of interactions like we have modified this work, say, done some kind of what is that interaction of amine groups, OH groups, there's some hydrogen bond to that. So then what happened after successful modifying this drug onto the carbon nanodots? We have also studied the drug release behavior at different pH. Then after that, then what we study the cell hybrid using a cancerous cell like S357 cell. Then he has studied this one. When he add this drug modified carbon dots into this cancerous cell, then this drug has been taken into the cells, and you can see that cell get die when you introduce this drug modified carbon dot. And also he has done some control experiment without this nanoparticle, and it was observed that this nanoparticle really helped to deliver these drugs effectively into the cell rather than only the drugs. So that was some interesting work we carried out using this carbon nanoparticles. And because this carbon nanoparticle is biocompatible with nature and its cell viability test is good, that's why it can be used. And also a lot of other nanoparticle and clusters of gold. If you counted number of gold atoms, you can develop a cluster. That also shows some patholuminescence properties. Less than 50 atoms of gold, less than 30 atoms of gold, you can develop also silver. 30 to 50 atoms of clusters you can synthesize. That can be used for this kind of applications also. So this I just want to conclude here that what I have shown to you, that the application of this nanoscience and nanomaterials for this biomedical application for development of electrochemical sensor, development of biosensors, development of optical sensor, and use in drug delivery applications.